Good afternoon. In the today lecture of computational botany, in the bioinformatics topic, we will discuss about the sequences used in bioinformatics. Here, the different sequences used in bioinformatics, that is genomic DNA, cDNA, that means complementary DNA, organelle DNA, ESTS means expressed sequence tags, and GSTS means gene sequence tag. So what is meant by sequence alignment? Sequence alignment means the matching or comparing of two sequences, that is amino acid or nucleic acid sequences, in one respective to another. In this, you can see here the first sequence is, that is ATGC. It compare with sequence 2, that is here in the other side of A there is gap. T matches with T and C with C and G match with A. So there is a base pair of sequence 1 and this is sequence 2. So here is a three possibilities, that is gap, match or mismatch. So the more match, that is more similarity in the sequence alignment. So where it takes place in those two sequences at least some similarities. So first one is genomic DNA. So genomic DNA compares an genome of an organism. This genome DNA is spread across the 64 chromosomes, leading to expression genetic traits. In the genomic DNA, it controls expression of various traits in an organism. So it contains the information of all genetic material in and of whole plant. So usually during DNA replication there is a recombination of genes bring about the changes in sequence leading to individual specific characteristic. So in the genomic DNA library, here the collection of clones containing all fragments of the genomic DNA of the organism in suitable vector. It contrasts the prokaryotic genome and eukaryotic genomes are very large and their genes generally carry the several introns. The genomic library contains DNA fragments represent in the entire genome of the organism. Then the genomic library made from the total nuclear DNA of an organism or species. In this, the DNA is cut into colonized size pieces or randomly as possible. In this, it is a process in which the samples can be analyzed or DNA samples can be analyzed. It can be extracted from the DNA from a given sample. तुम्ही एखाद्या प्राणी सॅम्पल किंवा ह्युमन ब्लडचे सॅम्पल घेऊन ते सॅम्पल ॲनालाइज करू शकता. In every normal cell there is a 23 pairs of chromosomes. In human blood there is a 23 pairs of chromosomes. In DNA, that is the DNA is double helical structure. You know all of that. In this process, when the DNA is double helical structure, so it can be unwinded. There, you know, the base pair of DNA, that is ATGC. So when the sample can inserted into the sequencing instrument for the sequence alignment. In sequencer, the each piece of DNA copied number of times, which creates the fragments. So the sequencer reads the DNA by using fragments or different color tags to each DNA base. In this process, the genomic DNA can be sequenced in different tools or sequence analyzer. Here the construction of genomic library. Here the genomic library means the collection of different, as you know, the library of books, there is the different books of different subjects. That means here the library of genomic library. Here the different genes are collected and stored in this library. Okay, so the genomic library contains whole genomic fragments including gene, exons and introns, gene promoter, introgenic DNA. Okay, so how you can be prepared genomic library? Okay, see here in first the genomic library. Okay, you can take a sample. In first diagram you can see the taking a sample of any DNA or DNA of plant sample or other, and you can insert it into the plasmid vector. Vector means the gene carrying vehicle which delivers the target genome into the host. So here is the host is, for example, E. coli. You can insert the genomic DNA into the host cell into E. coli. So you prepare a library of genomic DNA. So it can be stored in bacteria cells. Okay. So the samples can be inserted into the cleaved vector. That means the it can cleaved to insert the DNA sample into this vector. So in next stage you can insert it into the E. coli. In last one, insertion into the bacteria, and then isolation of this DNA for the collection of genomic library. You can see here in the test tube there is a library of different genomic DNA. Here the next one is cDNA, that means the complementary DNA. So the complementary DNA can be synthesized from the RNA, that means messenger RNA or mRNA. In this, the template in a reaction catalyzed by the enzyme reverse transcription. The complementary DNA is smaller than the genomic DNA. So it is a representation of only the coding region of gene expression of eukaryotic cell. Here the prokaryotic cell does not involve for the mechanism of splitting. So the DNA, the cDNA library only for the eukaryotic cell or organism. So the preparation of cDNA, here use of mRNA. That means the DNA can be synthesized from mRNA. Okay, that is exon. There is element of exon and introns, while exon are the coding regions and introns are the non-coding regions. So here the deletion of introns. So here is only exons can be take part in this for the preparation of complementary DNA So how can prepare the DNA from the mature mRNA? Here the cDNA is a DNA copy synthesized from the mRNA. The here use of reverse transcription enzymes for the preparation of DNA. That means it is a process of reverse transcriptions. That means RNA to DNA and DNA to RNA. That is transcription process. It takes place in the 3' to 5' end, and reverse transcription it takes place from 3' to 5' end. So the mRNA stand here is 3' to 5'. Here, see here, you can see here, this is mRNA strand. It is start from 3' to 5. In this, you can use the reverse transcription enzyme for the preparation of DNA. So, you can produce single stranded DNA, that is cDNA, that means complementary DNA. You can use only the coding region of mRNA, that means the exons which store the coding region of mRNA. In this, you can use the single stranded DNA. Here the prepare of cDNA which is single stranded. In this, we can hydrolyze or you can use the RNase enzyme for the cleavage of RNA, for which so you can prepare double stranded DNA. Okay. So you make a double stranded DNA from this. Okay. So hydrolyzing the mRNA strand or for using RNase enzyme or hydrolyzing. So in the last stage you can prepare the double stranded DNA, and it can be incubated with the termine transferase to add the single stranded tails. Okay, here is the double stranded cDNA which is only exons, that means the coding regions of an organism. Here the process of construction of cDNA library. In this synthesis of first and second cDNA strands. Here in previous slide we can discuss about the how you can be prepared the complementary DNA from mRNA strand. Okay. Here the methylation of cDNA at internal site and attached to the linker and adapter and restriction digestion. Here the ligation of DNA is to vector. That means this is a vector which can transfer the your sample DNA into the host cell. And last one is the amplification of cDNA clones in the for the preparation of cDNA library. Here the different uses of cDNA library. It is used in reverse genetic where the additional genomic information is of less use. Discovery of novel genes, cloning of full-length cDNA molecules for the vitro study of gene function. And this is a difference between the cDNA library and genomic DNA library. In the genomic library contains all genomic content of an organism, while in cDNA it contains only coding region of an organism. So the genomic library is much more complicated, and cDNA is simple. It genomic library it contains occurred prokaryotic and eukaryotic. So here required only in cDNA requires only eukaryotic cell. So we will stop here. And next lecture we can see the our next, that means the next sequences, genomic sequence tag and other two types. Okay, thank you.